Today we are going to talk about the pigmentation, and this includes lipofusion that we already studied in intracellular accumulation. Now in this video we will talk about the iron deposition, copper deposition, carbon deposition, and melanin accumulation and bilirubin accumulation. So from these iron, copper, they are dependent on, for example, if you are taking a lot of iron, that can cause accumulation of iron into different cells. Sometime, because of some metabolic error, there will be accumulation of iron, but it could be exogenous, it could be because of some endogenous abnormalities. A copper, the same, because of some enzymatic defect, because of some genetical defect, like in case of Wilson disease, there will be ATP B7 gene mutation that can cause accumulation of the copper in liver and different other organs. And about the melanin and bilirubin, these are normally produced by our body, and once their excretion process is affected or production is become more, especially if you talk about the bilirubin, then in case of the hemolytic anemia, there will be excessive production of the bilirubin than that of the excretion, so there will be accumulation of the bilirubin. Or the melanin is same sometime, because of some melanoma, there will be excessive production of melanin, and these melanin start to accumulate in your nails, in your eyes, and in skin as well. Pigmentation. Anthracothic pigmentation. So they are saying that which is the carbonaceous debris from the urban living or the cigarette smoking. This causes accumulation of the carbon. Endogenous pigments including melanin and bilirubin. So this two image we can see in the image one, it's a liver, and it's it turned into black because of accumulation of the iron. So it's the hemochromatosis. While the second image, they're showing a ring around the eye, or you can say at the limbus region, and this is accumulation of the copper in the cornea, and this is called as the Kasher-Fleischer ring. So I will in detail description I'll tell you about what are these problems. So the first one is accumulation of iron. So in this image you can see that this is what in the heart on gross image, and this is the microscopic image. So in gross image you can see that this chamber is completely dilated, and then this one. So this is the right ventricle, and this is the left ventricle. So because of accumulation of iron, there will be dilation of chamber. So iron accumulation can also cause dilated cardiomyopathy. And the excess iron accumulation, when it leads to symptoms or it leads to certain problems, that is called as hemochromatosis. So if I want to take a sample from this myocardium and I want to study here, I can see that these are the cardiomyocytes, cardiac muscles, and I have seen some bluish depositions. So all these blue depositions, they are nothing but they are iron accumulations. So this is the microscopic image of iron accumulation, and this one is the dilated cardiomyopathy as a result of hemochromatosis. So let's go to the description. In the description, they are saying that this is the iron accumulation in the heart myocytes, also called as hemochromatosis or hemosiderosis or hemochromatosis. Usually hemochromatosis is a dangerous condition because they can cause different structural and functional abnormalities in the organ, while hemosiderosis can lead to less likely the development of symptoms. The stain that used is the Prussian blue or red stain. The name is blue and the appearance also blue. So if someone asks that a patient died because of cardiomyopathy, because of dilation of the heart, and on autopsy or microscopic finding, when you stain that sample, you found some blue color depositions. What is that deposition? You should answer. It should be iron. And what is the stain? That is the Prussian blue or pearl stain. The accumulation is iron. Color is blue. Organ is heart. So location of the iron accumulation, all these blue, they are the accumulation. Normal cardiac muscles. So in this area you can see these are the normal cardiac muscles, In dilated chamber is the first image. Here you can see this one is the dilated left ventricle. Let's go for the next image. This is the same image, but the difference is the staining. What is the difference? These are the again cardio muscle cells. All these are cardiac muscles. These are the nuclei. All these are the nuclei. And just near the nuclei there is a deposition inside. These are the intracellular depositions. Last time we studied that if it is yellow brown, we studied that would be lipofusion, because lipofusion related to free radical injury, old age, and perinuclear, just near the nucleus. Here you can also see that this substance is somewhat about near the nucleus, but this is not associated with the old age and not associated with the free radical injury. So these depositions are golden brown in color. So golden brown intracellular, if it is in the heart and in blood there is a high level of iron, then it gives some points toward the hemochromatosis or hemochromatosis. So this is again the accumulation of iron in the heart. So description, they are saying that iron accumulation in the heart myocytes. The staining here is different. In previous it was blue, so that was Prussian blue, but here it's somewhat golden brown. So golden brown, if it is iron, then the stain should be hematoxylin and Ihsm staining. The organ will be the heart. Find the accumulation of iron. These are the iron accumulations. Normal cardiac myocytes. These are the myocytes with the nucleus. Name the stain used is the hematoxylin and Ihsm staining. The next image is a gross image of liver. So they're showing an area where we can see iron accumulation, and the whole liver become, it's a little bit dark. So this is what the gross image of the liver with loaded with iron. So hemochromatosis. So this is they are saying. This is the iron accumulation in the liver. The disease is hemochromatosis or hemochromatosis. It's a gross image, so no staining. Iron accumulation, brown black on gross examination. And organ is liver. Name the condition is hemochromatosis or hemochromatosis. Next one is the histological image of the the liver. So here we can see that normally the structure of liver is like some of this. Inside the central vein, these are the portal tracts, hexagonal, and at the end there's a portal tract. Portal tract including the hepatic portal vein, hepatic artery, and bile duct system. So these are the sinusoids. The cells near the portal called periportal, and the cells near the central vein, they are called the centrilobular. So these cells are the centrilobular, and if this one is the portal tract, then these cells are the periportal. And all this blue, they are the iron accumulation by Prussian blue staining. So the central vein, portal tract, preportal cell, centrilobular area, and iron accumulation. Next one is the same, but the staining is different. It's easy now. So these are the hepatic cells, hepatocytes, and this is what the nucleus, cytoplasm. So inside the cytoplasm there is brownish deposition, that is iron. All these cells, they are loaded with iron intracellularly, and it's a golden brown in color. So iron accumulation in the lungs, in the liver, and is the histological image. So they're saying that iron accumulation in the hepatocytes, staining is hematoxylin and Ihsm, accumulating substance is iron, color brown golden, liver is the organ, location of iron deposition in the hepatocytes, all these are the location, intracellularly, hepatocytes, so these are the hepatocytes, one hepatocyte, two hepatocyte, all these are the hepatocytes. Name the pathology and the stain. The pathology may be hemochromatosis or hemochromatosis, and the stain is H and E stain. Next one is by Prussian blue, all these are the hepatocytes. Okay, and these are the iron accumulations by Prussian blue staining. So this is like iron accumulation in the hepatocytes, stained by Prussian blue. So iron accumulation in the hepatocyte, stain is Prussian blue. Accumulating substance is iron, color blue, organ is the liver. Next image is a gross image of lungs. So this is the lungs, trachea, bronchi, left and right. Then divided into fine bronchioles. This is the fissure. If you compare this area with this area, you can see that the color is here. It's somewhat more brown. This one is a little bit pale. So this is what we call as the brown induration of lungs. And you all know that what does brown induration mean? Brown induration means when there is in the alveoli excess accumulation of iron into the macrophages. So these are the macrophages loaded with iron. And this macrophage is called as hemocidal laden macrophage. And the color of iron is brown, so it gives rise to brown induration of lungs. Upon histology, you can see that this is the image of lungs, the alveolar space. This is again the alveolar space. Some alveolar space are loaded with some brownish color cells, as this alveoli is again loaded. And the septum is thick, the septum becomes so thick. Here also, the septum is so thick. So there are some intracellular, or you can say intra-alveolar depositions, as well as in the septal deposition. So these are the septum. In the septum, macrophages again loaded with iron, and in the alveolar space, macrophages is loaded with iron. So these are the macrophages. If I took this out and try to make here, so this one will be the shape of macrophage. This is the nucleus, and it will be the iron accumulation, because this is engulfing a lot of RBCs. So there will be macrophage with iron and hemocidin-laden macrophages inside the alveolar space and in the interstitium or in the septum. And these are the alveolar cells, pneumocyte type 1, and these are the septal cells. So this is again brown induration of lungs, or that the cause of this is venous congestion of lungs. So this is hemochromatosis or hemochromatosis. The next image is, and the stain here, if I talk about the stain, then the stain will be hematoxylin and Ihsm stain. Okay, let's figure out that one. So it's iron accumulation in the alveoli in a septal space. It's called a brown induration of lungs. Stain is hematoxylin and Ihsm, iron accumulation, golden brown in the organist lungs. So this one is very, very important, very beautiful image. So here we can see that this one is the pupil, and here is in the cornea, at the limbal area, there is a circular ring, brownish. This is called as Kessler-Fleischer ring, and this is specific for copper accumulation. Which layer of the cornea is in the Descemet's membrane? So Kessler-Fleischer ring, a golden brown color, no stain, it gives color by itself. And in the Descemet's membrane, and the disease where we can find some accumulation is Wilson disease. Let's go for the detail of this image. So it's a copper accumulation in the eyes is the Descemet's membrane of the cornea, and the ring is called Kessler-Fleischer ring. The disease is Wilson disease. It's a gross image. Accumulation is copper, color brown, stain that can be used histologically for the copper. In this there is no stain used, but if you take a sample of liver, or it's also accumulated in the brain in the basal ganglion, and you took a sample from that and try to see the copper, then the stain you must use for the staining of copper is the rhodamine and robionic acid. So you must remember that rhodamine and robionic acid, these two stains, they are used for the copper identification. Organ is eye, also accumulate in the brain, in the basal ganglion, and in the liver, also in the hepatocytes. If it accumulates in the brain, that leads to chorea or some abnormal movement, lack of coordination of the movements, and in the liver, it causes cirrhosis of the liver. Next is the accumulation of black substance into the lungs, that is the carbon accumulation. So here we can see this is the lungs, one on the third side, and in between the two lungs, this one is the heart, and these are the ribs. So all these black depositions, they are the anthracotic carbon or carbon deposition. If you take a sample and study on the histologically, you can find that these black depositions in the alveoli, into the alveolar space, in the bronchial cells. So all these are the carbon depositions, and these are the this is the macrophage. You can see like this one, and nucleus like this. This one again a macrophage, here macrophage, and these two macrophages, they are loaded with carbon. So this is macrophage laden with carbon. This is somewhat like a neutrophil. And these are the blood vessels. And if you try to find the lining of epithelial cell, you need to zoom in. You can find this one. I can see it's a squamous cell. It's the lining of alveoli. So what they're saying that it's an anthracosis of the lungs, mean accumulation of carbon. Anthracotic pigment is a carbonaceous material that accumulates in the lungs of a smoker, coal worker, person living in a polluted environment such as large cities. The black anthracotic pigment typically accumulates in the pleural lymphocytes, and microscopically a finely stippled black appearance found in the interstitium, also alveolar macrophages, and this image is hematoxylin and eosin stained. Next is a very important also. So here is the sample or the microscopic or the histological image of the skin, where we can see this is what the stratum corneum, stratum granulosum, stratum spinosum, and the last one is the stratum basale, the last layer. So if you know that in the last layer we have the melanocytes. So melanocytes are present in the stratum basale, and these melanocytes are loaded with melanin, and melanin, it appears like a brownish color on H&E staining. So they are saying that these cells are keratinocytes, usually present in all layers except the stratum corneum. Melanocytes present only in the stratum basale, loaded with melanin, and these are the keratinocytes of the stratum spinosum or stratum basale. They possess, this one is a melanocyte, it has a dendrites, and these melanin will go like this and it will go up, and on the stratum spinosum they start to enter into the keratinocytes and give a pigmentation to the skin. And these are the keratin pearl, these are all the keratin fibers, and they don't have the nuclei because they are only the fibers, collagen like fibers. So stratum corneum is only a layer of keratin. This image, if you want to look, you can see that this is what the basale, stratum basale, and stratum spinosum, granulosum, and stratum corneum. Stratum corneum is only the fibers, granulosum, spinosum, and basale. And this is the dermis. And this all layers are the epidermis. So we can see that here a lot of brown pigmentation. So it's nothing but the melanin accumulation. Melanin accumulation in excess. And the stain using here is hematoxylin and eosin. They give rise brownish color to the melanin. So let's see the description. It's endogenous melanin accumulation in the skin. The stain is hematoxylin and eosin. Accumulation is melanin, color is brownish, accumulate into the skin. But you also must need to remember where else it's accumulate. Location of melanin in the layer of skin, I already showed you. This is the location. Melanin producing cell, they are the melanocytes, and function of melanin gives pigmentation to your skin, iris, eye, brain, substantia nigra, in eye, at the iris, skin, brain, substantia nigra, all these are the areas. Nail also, it gives pigmentation. Here also, and nails. Next one is again the same, but the staining is different. So this is the epidermis, stratum corneum, stratum granulosum, stratum spinosum, and stratum basale. So at the basal layer, highly black depositions. So these are not brown. So could it be melanin? Yes, it's a melanin. So this is the melanin accumulation, endogenous melanin accumulation, but the staining is different here. If it is hematoxylin eosin, it should be like brownish in color, but here it's not brown, it's a black in color. So this is what the Mason-Fontana staining. So they are saying that it's a melanin accumulation in the skin at the basal layer. Staining is Mason-Fontana staining. Accumulation in melanin, color black. Organ is the skin. Location of deposition is the stratum basale. Organs where else it can deposit: brain, eyes, hair and nail. Layers where melanocyte present. Which layer is the stratum basale, the basal layer. The next image is the bile accumulation, because you can see this is the liver, because liver is the organ which take bile and excrete the bile. So bile has a color of greenish color, so we can find all these greenish areas, they are the bile accumulation. It means that if the liver is taking bile from the blood, it needs to drain out. So if there is a problem with the excretion, then bile start to accumulate, and this condition is called as the biliary stasis. Bile stay into the liver, and this marked arrow they are showing a thrombus into the portal vein. So if there is obstruction, there will be accumulation of bile, and the bile give rise to a greenish color. They will be enlarged in the size of liver. There will be jaundice of the skin, submucosa or the mucosa, along with the cornea or the conjunctiva. Okay, so what they are saying in histology is the same, this one. So all these are the bile accumulations, bile stasis in the bile duct, small lacteals, and these are the hepatocytes. So they are saying that it's a bile stasis or the cholestasis within the cirrhotic liver. Cholestasis is the accumulation of bile, an endogenous pigment produced by the liver. Its accumulation can result from many processes, including bile duct, the obstruction of the bile duct. Grossly, cholestasis will result in the greenish discoloration of the liver. The arrow indicates a portal vein thrombosis. Microscopically, bile is a globular and yellow-green in color. The arrows indicate the accumulation of the bile, these arrows. And the staining is hematoxylin and eosin. They appear slightly reddish in color on histology, and gross is like a yellow greenish in color. This is again the same image, but little bit description. So these are the hepatocytes, and this is the bile accumulation. So what they are saying, it's endogenous bile accumulation in the liver. Stain is hematoxylin and eosin. Accumulation is bile, color is reddish, organ is liver. Location of bile accumulation, biliary coli. Types of bile, there are two types: conjugate and unconjugate, or we can say direct and indirect bile. So this was something about the pigmentation and accumulation of different sort of pigments at different organs.